Welcome to this explainer. You know, whenever we think about reproduction, it's so easy to picture the male gamete, the sperm, as this independent, self-made hero on a solitary quest to reach the ovum. But when you look at the actual physiological data, a totally different story emerges. Today, we're going to reframe male reproductive physiology as an epic hero's journey. But rather than just focusing on the hero itself, we're zooming in entirely on the grueling, highly engineered training environments that actually make fertilization possible in the first place. Because without these environments, our hero wouldn't even make it past the starting line.
Here's our roadmap for the journey. We're going to look at the ultimate guardians known as Sertoli cells, go inside the germinal epithelium, head to boot camp for sperm maturation, face the final trial of capacitation, and then compare those two trials side by side.
First up, the ultimate guardians, the Sertoli cells. We are starting our story right in the nursery.
Before a hero can go on a quest, they obviously need to survive infancy. To give you a sense of scale and time here, the entire developmental period for a sperm cell, which is called spermatogenesis, takes about 74 days. Over those 2.5 months, a single primitive germinal cell divides and transforms, eventually giving rise to 512 mature sperm cells. That is a massive amount of biological manufacturing happening around the clock, and it requires some seriously intense, dedicated support.
Enter the Sertoli cells. The key term to remember here is agerminal, meaning these are not the germ cells that actually become the sperm. They are the essential support staff. Residing in the seminiferous tubules, they're absolutely crucial because they provide the mechanical support, the protection, and the high glycogen nutrition that these maturing gametes need. They literally envelop the developing cells to keep them alive.
But they aren't just physical shields, they are highly active chemical factories. These cells are biochemical powerhouses. You can easily remember their complex endocrine functions using the MAAAI mnemonic. That's MAAAI. First, they secrete Mullerian-inhibiting substance, which stops the development of female sex organs. Then androgen-binding protein to keep local testosterone levels super high. They also have aromatase to convert androgens into estrogen. And finally, they secrete both activin to stimulate follicle-stimulating hormone and inhibin to suppress it. They are meticulously regulating the local hormonal environment to ensure perfect training conditions.
Part 2. Inside the germinal epithelium. We know what these cells do chemically, but let's see how this plays out geographically. When you look closely at the microenvironment of the testicle, you see these towering Sertoli cells completely enveloping the much smaller developing germ cells. They span the entire thickness of the epithelium. They're acting as a living barrier and a nursemaid all rolled into one. It's an incredibly intimate structural relationship. And to achieve this level of protection, Sertoli cells form a tight, zipper-like blood-testis barrier. This barrier physically isolates the developing sperm. It stops random proteins from entering, but more importantly, it prevents antigenic substances from escaping into the bloodstream. Why does that matter? Well, without this strict isolation zone, the body's own immune system wouldn't recognize these newly formed cells and would actually attack them as foreign invaders. The Sertoli cells are literally hiding the hero from the body's own defenses. Moving to part 3. Boot camp, or sperm maturation. Having successfully left the safety of the nursery, our biological hero enters a high-stakes boot camp phase. This maturation takes place in a coiled tube called the epididymis. Now, when the sperm first arrive here, they are completely non-motile. They can't swim, and they certainly can't fertilize an ovum. But over the next 18 to 24 hours, bathed in special nutrient fluids and enzymes, they undergo a massive transformation. They acquire the potential for motility and the capability to fertilize. But here is the fascinating catch. Even though they are now fully capable of swimming, chemical inhibitors in the fluid strategically hold them back. They're fully equipped, engines are revving, but the parking brake is firmly pulled up. They won't actually swim until after ejaculation. Which brings us to part 4, the final trial, known as capacitation. After ejaculation, the environment changes completely. The hero has left the male body and entered the female genital tract. And this is not just some passive waiting period. Capacitation is a rigorous 1 to 10 hour biological activation process driven entirely by the female genital tract. This is the phase where the training wheels finally come off. It takes the sperm from being merely capable, remember that parking brake from the epididymis, to being fully activated and physically prepared to penetrate the ovum. The sequence of events here is beautifully elegant, and it unfolds in 3 distinct steps. First, the uterine and fallopian tube fluids physically wash away those inhibitory factors from the epididymal boot camp. The parking brake is officially released. Second, the sperm sheds cholesterol vesicles that were covering its head, which is called the acrosome. Think of it as shedding its chemical payload helmet. This loss of cholesterol makes the membrane weaker, which sounds bad, but it's absolutely essential for releasing enzymes later. And finally, this weakened membrane becomes highly permeable to calcium ions. This sudden calcium influx is the spark that creates a powerful hyperactive whiplash motion in the flagellum and primes the acrosome to release its penetrating enzymes. Section 5, Maturation versus Capacitation. Because these 2 distinct trials are so critical, but honestly pretty easy to confuse, we need to draw a stark contrast between them right now to cement your understanding. Looking at them side by side reveals a really stark difference. First, think about the site and the time. Maturation is a slower 18 to 24-hour preparatory phase happening in the male's epididymis. Capacitation, on the other hand, is a rapid 1 to 10-hour activation phase entirely in the female genital tract. And the outcome is totally different too. Maturation gives you a sperm that is capable but completely inhibited. Capacitation gives you a fully active, whiplashing sperm ready for the acrosomal reaction. Basically, one builds the potential, and the other unleashes it. Which brings us to the end of our explainer, and leaves us with a bit of a philosophical question. We've seen how the Sertoli cells hide, feed, and chemically sculpt the sperm. We've seen how the epididymis grants it potential while holding it back. And we've seen how the female reproductive tract entirely re-engineers its membrane and movement through capacitation. So, considering the incredible, complex environments that do absolutely all the heavy lifting to make fertilization possible, who are the real heroes of this biological journey? Is it the vessel that arrives at the destination, or the environments that built it along the way? Something to think about. Thanks for joining me on this explainer, and keep questioning what you learn.